Good morning, all the participants and our eminent speaker, Dr. Padmanabhan. I'm Ujjwala Shinde, HOD of Pharmaceutics Department, Bombay College of Pharmacy. I welcome you for session 8 of day 3. It gives me an immense pleasure to introduce Dr. Padmanabhan, who is ex-Deputy General Manager, Radiopharmaceuticals, Quality Control at Board of Radiation and Isotope Technology. He has vast experience of preparation and quality control of labeled compound, namely deuterated hydrogen and carbon-14. He is the recipient of 4 DA Group Achievement Awards. He has got 36 publications to his credit in national and international journals. He served as PhD examiner for Lucknow, Saurashtra, and South Gujarat University. In addition to this, I am very happy to share with you all that Bombay College of Pharmacy has long association with Dr. Padmanabhan in various research projects. With this brief introduction, I welcome Dr. Padmanabhan, who is going to talk on quality control of radiopharmaceuticals. Over to you, Dr. Padmanabhan.
Okay, thank you very much, madam, for the introduction. Yeah. And good morning to everybody. Am I audible to you, all of you?
Yes, sir. Yes.
Okay. Now, what about the presentation is not seen in my 6 slides, or I will not operate through my presentation. That PPT. Hello.
Hello.
The PPT is you will be showing on the thing, or I will have to open the. I can open.
Yes, sir. You can also share. You can also share.
That's what I do. One. Because last time they have.
Yes, sir.
Yeah, one.
Yes. Yes.
Let us open. I will operate. Now you are able to see the slides? Hello?
Yes, sir. Hello? Yes, sir.
You are able to
see your slides.
Okay. First of all, I would like to thank the organizers and the management for giving me this opportunity, especially to Dr. Vaishali Shishwath for me to give the invitation. Thank you very much, madam. Okay.
Thank you, sir. Thank you. Our pleasure, sir.
Yeah, thank you very much. And now today I have chosen the topic quality control of radiopharmaceuticals. Now this voice is okay, or will I have to slightly loudly I have to stop? That you please inform me, because online some of the now you are audible to everybody.
Yes, sir. Your voice
is
okay.
Yes, sir.
Okay. If it is not clear, please tell me. You can interrupt in between also. Okay. Hello.
Yes, sir. Yes, sir. Okay. Yeah, sir, you're audible. You can continue.
Okay. Quality control of radiopharmaceuticals. See, what we do, the quality control of pharmaceuticals and radiopharmaceuticals almost same, except for few tests, it is slightly different. That special test thing we are going to see this topic. Okay, why the special tests are carried out? Because of the radioactivity component present in the radiopharmaceuticals. That is one point. What we do in the quality control department for the laboratories, we carry out some battery of tests, okay, for the product, and to ensure the product is safe and efficacy is good and safe to use in human administration for the pharmaceutical as well as for the radiopharmaceutical. That is the purpose of quality control department is there in the any production unit. Now, coming to the quality control, in this topic, what I am going to cover this thing is, since the radiopharmaceutical is slightly different from the pharmaceuticals, mainly the radioactive component is involved. So, what are the things, safety aspects I am going to cover it, and what way we have to safely handle the radioactivity. What are the special tests we carry out for radiopharmaceuticals, in addition to the general tests for pharmaceuticals we carry out. Then, what are the special instruments for carrying out that special test we are going to see, and in addition to that, what are the precautions we have to take it, we have to see, and other various aspects who are all governing the thing, what is that? Regulatory aspects also we are going to see in this talk. Now, coming to the quality control topic. First of all, I would like to start with a small general introduction about the radioisotope, radioactivity, specificity of like all the important terms I want to introduce to you so that you can understand in a better way. Otherwise, what happens suddenly if I go to some radiochemical purity or radioactivity concentration will be a thing. You may be knowing it. Most of the faculty members are new, I heard from the professors. At the same time, some of the terms you would have studied in college or school level, you would have forgotten also. I want to refresh it first. Then you will be able to appreciate, understand in a better way or grasp in a better way. That's why I start with small introduction, general introduction with these small terms. First one is radioisotopes. This I did not much explain on the radioisotopes. This isotopes, all of you know it already. Isotopes are the atoms of the same element having the same atomic number but different in mass numbers are called isotopes. This you would have studied. But what are radioisotopes? Radioisotopes are nothing but isotopes having the radioactivity are called radioisotopes. And they are also called as radionuclides. So radioisotopes, radionuclides, the terms often it will be using in this topic. Therefore, this is you have to understand very nicely about the radioisotopes. And there are the three isotopes. Best example, if I want to give, hydrogen element is having three isotopes. One is protium, another is deuterium, another is tritium. Out of this, protium and deuterium are stable isotope, and tritium is a radioactive isotope. Radioactive means it is not stable. It goes on give some energies like alpha, beta, gamma. Type of any one of this energy will be giving in order to stabilize that element. So that isotope will be stabilized by giving out excess energy. So in this case, there are two types. One is stable isotopes and unstable isotopes. Unstable nuclei are called radioisotopes or radionuclides. Then coming to the types of radioisotopes. Once you understand the radioisotopes, you are supposed to know how many types of radioisotopes are there. And here we are going to see different types of isotopes which are normally used in the radiopharmaceutical preparation. Okay. So now coming to the types, one is alpha emitting isotopes, beta emitters, gamma emitter, positron emitters. These are the main things we are using in the radiopharmaceutical preparation. And out of which alpha, I am not going to talk much about alpha emitters, that the uses in the radiopharmaceutical are highly negligible. Very less amount of radiopharmaceuticals are used, very less negligible amount of things. In India, not much is used. The alpha emitters are not much used. Whereas beta, gamma, and positrons are very common, and out of which the positron emission isotope is becoming very popular in this thing, where it is produced in cyclotron. That we will see in later. First, coming to alpha emitter, why this is not much useful in the thing? Because the range is very small. Generally, radioisotopes for the preparation of radiopharmaceuticals, we are using it. That is mainly for diagnosis purpose we are using it. The emission should come out. We are finding out the emission from the body. When it is injected into the body, it gives out the emission that alpha is emitting the rays or beta is emitting the rays. That rays has to be captured by the camera. But the camera, since the range is very, very small, so it is not possible to magnify or capture the photo of the alpha. Therefore, alpha emitters not much useful for the diagnosis, but to some extent, some therapeutical use, some of the things are used. That is radium chloride is the one best example I can tell. Radium-223 chloride is used. Coming to beta emitters, a lot of beta emitters are there. That iodine-131 we are going to see in this. Only one isotope I have taken. Gamma emitters, technetium-99 and one thing we are going to see. And positron emitter F18, that the very popular radioisotope we are going to see in this radiopharmaceutical. And coming to radioactivity, this is the basic thing. Radioactivity is the phenomenon in which what happens, that spontaneous emission is coming from the radioactive atoms or unstable nuclei. So that is known as radioactivity. This is very, very important in terms of this particular quality control. The units of radioactivity must have studied millicurie. Millicurie curie and microcurie, etc. are there. But SI system is units are becquerel, megabecquerel, and gigabecquerel, terabecquerel, etc. is used. What is 1 becquerel? 1 becquerel is nothing but 1 disintegration per second. Later slides also we are explaining radioactivity. When it comes to some other importance radioactivity, I'm going to explain further. Now coming to specific activity, this term is very very important as far as radiopharmaceutical is concerned. Even though we are not going to test, test is not carried out for specific activity by our QC department, but this is very important. Many people are getting.
Hello.
Yeah.
Sorry, Dr.
Yeah.
Padmanabhan sir.
Yeah,
Your audio is not that clear, sir. Can you switch off the video? Maybe then the audio will be more clear. So.
Pardon? Video?
Yeah. Can you switch off the video? Maybe then we'll be able to hear you clear.
Oh my! Switch off the video. 1 minute. I have to escape. 1 minute. Video. 1 minute.
Yes, sir.
Video shall I switch off? Now it's okay. Hello.
Yes, sir.
Now switch. It's okay. Hello.
Yes, sir. It is all right.
It is all right.
You can switch off the video, sir, and you can talk.
Hey, that's what I'm seeing. How to switch off only this. 1 minute. The camera is.
So it was switched off before. Now you have again switched on. That's fine,
Madam?
sir. Yes.
It's okay now.
Yes, sir.
1 minute. Again on. Now it is off.
Yes.
Hello. Turn the camera. It is off only, no? Hello.
Sorry, I think you have switched on the video again. Please switch it off.
Now, so I switched off. Now it is okay.
Yes, sir.
Now I.
Please continue. Thank you.
1 minute. Okay. So now it is clear. Specific activity.
Yes.
Now, the term specific is very important thing because many students, research students and professors are getting confused with the specific activity. Specific activity is nothing but it is defined as the radioactivity per unit mass. Okay. It is expressed as the millicurie per milligram or curie per gram or curie per milligram also it can be there or megabecquerel per milligram or gigabecquerel per gram. Any unit weight you have to take it. In what it indicates, specific activity indicates the abundance of radioactive atoms in unit mass. This is very important. That's why I put one more slide for this. Example for the thing, 10 millicurie of any compound, you take 10 millicurie per milligram, 100 millicurie per milligram. What is the difference? In 1 milligram, there are a lot of activities there. In 1 milligram, 10 millicurie is there. The difference is, in the case of second 100 millicurie per milligram, the radioactive atoms present in a milligram is more. Number of radioactive atoms are more. It also indicates the non-radioactive atoms are less in this case. So, what is the graphical, this pictorial representation I have made it. You are able to see that the slides, there are 2 blocks I have made it, 2 squares, almost having let us assume they are having the same weight. You are able to see the slide? Hello?
We are able to see the slide.
Yes, sir.
This X indicates radioactive atoms, whereas 0 indicates non-radioactive atoms. In the same square, you see the other square, you see the low specific activity. X is radioactive atoms are very much less, whereas 0 non-radioactive atoms are more. But the given weight is same. Both the weights are same, or both the squares of area is same. In one case, the activity is the number of radioactive atoms are more, the number of radioactive atoms are low. So in what way it is going to impact the quality control test, or in what way it is useful for the radiopharmaceutical? I will be explaining when it comes to the particular slide. Okay. So this, I think this is clear. Radioactivity is radioactive number of radioactive atoms will be more. Activity per milligram. Okay, that is per unit mass. It explains. That means high specific activity means number of radioactive atoms are more in a given unit mass, and low specific activity means number of radioactive atoms are present in the particular unit mass. That is the difference between the specific activity, the high specific activity, low specific activity, and radioactivity. even the radioactivity will become off, but the specific activity will never decrease. That is the thing one should understand. Coming to the half-life, that is very important phenomenon. Half-life that is going to be useful in understanding the quality control test. Half-life of any radioactive element will have half-life. What is the half-life is? The activity will become half of its activity after its half-life period. That means each and every radioisotope has got the half-life. Example I given, technetium 99M has got one radioisotope, which is very useful in radiopharmaceutical, has got 6 hours of half-life. That means 10 millicurie if you take it, 10 millicurie of radioactivity if you take it, what will happen? After one half-life period, it will become 5 millicurie. 10 millicurie will become 5 millicurie after 6 hours. That is technetium 99. At the same time, C14, if you see the half-life, it is having 5,730 years. What is the C14? Same 10 millicurie if you take it, and after 5,730 years, it becomes 5 millicurie. So what do you understand from this? So one is emitting the rate of decay is very high. Within 6 hours, half of the radioactive atoms are decaying. Wherein C14, since the half-life is so high, it will decay at the slower rate. Slowly, one atom will decay, second atom will decay after some time, something like that, it will decay. So that is the difference between the half-life. The half-life is short, the decay rate is high. The half-life period is high, the decay rate is very slow. That's what we have to derive. And in the case of radiopharmacy, why we have to study? The radioisotope where the short half-lives are there, then it is preferable for the radiopharmaceutical preparation. Of course, the exception case is there. C14, we have made one radiopharmaceutical using C14 also. That urea capsule we are using for Helicobacter pylori detection, we are using it. But it has got the half-life, even though it is having the half-life of 5,000 something, but its shelf life. Shelf life, we are going to see that time we will see that. Explain what is the shelf life of radiopharmaceuticals. Now coming to radiopharmaceuticals. It is very simple. Pharmaceutical formulations. What are radiopharmaceutical? Pharmaceutical formulation containing radioisotopes which are intended to be used for either for diagnosis or for therapy. Okay. So what is the thing? Again, in the layman language, see any organic molecule, if you take, or inorganic molecule, take it, which you incorporate radioisotope in it, then it can become a radiopharmaceutical. Then formulated thing form will be a pharmaceutical, radiopharmaceutical. What is very important? Any organic molecule or inorganic molecule, you incorporate the radioactivity in it. Then, if they are having the pharmaceutical properties, definitely it is called as radiopharmaceutical. In India, who is manufacturing the radiopharmaceutical in India? Only one laboratory, that is Board of Radiation Isotope Technology, that is the Department of Atomic Energy, produces all types of radiopharmaceuticals, and it supplies to more than across the country, more than 150 radiopharmaceuticals in the hospital, nuclear medicine hospital, we are supplying. So the BRIC is the only source of this radiopharmaceutical. Otherwise, people are now that private hospitals are importing the thing. As far as the production is concerned, only India, British manufacturing, and of course BRC is also some extent they are manufacturing. BRC, BRIC, and Radiation Medicine Center at Parel. That is also part of BRC. They are producing F18 labeled radiopharmaceutical in collaboration with BRIC. So both are involved. Radiation Medicine Center as well as BRIC. Wherever the manufacturing is there, always BRIC is only the selling source for the BE department. Coming to the difference between the pharmaceutical and radiopharmaceutical, very interesting to know the thing. So pharmaceutical, what way is different from the normal medicines? First of all, a very distinct point is have a radioactive component in pharmaceutical. Radiopharmaceutical have radioactive component is there. The pharmaceutical there is no radioactive component. Then how have short of shelf life. Then the expiry date when compared to the pharmaceutical will have months to years that expiry date will be there. But in this case, it will be in hours. Some of the radiopharmacy you have to use within 1 hour, within half an hour you have to use it. And some of the products are maximum range is say 25 days, 30 days, maximum expiry date is not more than that. But except on the exceptional cases, C14 urea capsule have around 6 months of expiry date. Then, since they are having the shelf life, the expiry date is very small. Some of the things are having 1 hour or half an hour. What immediately we have to use it. So they should be prepared. What we do, with certain radiopharmaceuticals are prepared and dispatched immediately to the doctor's end. Within half an hour, 1 hour, we are supplying to them. So immediately they will not use it. Otherwise, it will be decomposing because of decaying nature. The expiry date is very short. Now, pharmaceuticals mainly, the pharmaceuticals are generally used for the therapeutical purpose. They are used, whereas majority of radiopharmaceuticals used for diagnosis, just opposite to pharmaceuticals. Now the therapy use is slightly increasing. Otherwise, majority of the radiopharmaceuticals are mainly used for the diagnosis. Then, in addition to this properties, the safety of the operator and final user should be ensured. In this case, the safety, where the safety comes, the safety component is coming because of the radiation involved. That is, radioisotope will emit rays. That rays should be, we have to protect we should be guarded with the rays, that we should not get the radiation to the operator. Those who are using, the doctors, those who are injecting, or those who are handling the technician, they should not get the radiation. Okay. At the same time, user also should not get more dose to the thing. For that, the safety is also ensured taken care by the radiopharmaceuticals. Then coming to the production of this idea I'm giving, we are not got the topic of this one. Peripheral things we are just touching. Production of radioisotopes. When we come to the radiopharmaceutical topic, we should know where the radioisotopes are used. There are three sources are there. One is research nuclear reactors, that is nuclear reactors. Second is cyclotron. Third is generators. People, general idea about when pharmaceutical people, even pharmaceutical professor, even Andre, I'm not particularly telling the thing, even chemistry professor, when they ask where the radioisotopes are, many people, they don't know. And nuclear reactors, if I say nuclear reactors, many the people think that nuclear reactors meant for production of power production of electricity. It is not like that. It is research reactors. Research reactors design is different from the power reactors. So in the research reactors, we are getting the thing. Here, what is done is the neutrons are bombarded to the target material. The target material is converted into radioactive material. That is the basic things we are doing. Okay. That is, the target material is exposed, irradiated with neutrons. Then the target material is converted into, and then purification, everything is done. The processing, purification, everything is done. Then we are getting the pure isotopes. And then coming to cyclotron. Cyclotron is the particle accelerator. When we accelerate a particular particle and bombard with the target material, we are getting the radioisotopes. This way also one can do. When compared with nuclear, what is the difference between nuclear reactors and cyclotron? One is particle accelerator, where other one is a nuclear reactor, where the nuclear reaction is taking place. Neutron is bombarded with the target is bombarded with nuclear neutrons. Here in research reactors, you can prepare large quantity of radioactive atoms. Whereas in this case, cyclotron, the quantity production will be less, and special isotopes can be prepared in the cyclotron, only small quantity. And coming to generators, the device of generator is also a source of radioisotopes, pure radioisotopes. That I will explain generators. Now, coming to this slide indicates that Dhruva reactor, which is in Trombay, in BRC. There are two reactors shown here, one dome type, another is rectangular building is seen. There the chimney is seen, that is the Dhruva reactor, that is the new reactor. One side of the dome-shaped reactor is now it is closed. Previously, we are getting the radioisotopes from the two reactors. Now we are getting from the Dhruva reactor. Now this is dome-shaped, that is called a Cyrus reactor. It is closed down. It is because of aging problem. So many decades it served. Now the new reactor is the Dhruva reactor. This is the only research reactor available for our radioisotope program, where the many radioisotopes are produced in this reactor, processed, and we are getting pure radioisotope. Then we bread Washi complex, we process and make the radiopharmaceuticals. Now this slide is clear, seen to you clearly, all of you. Hello. Hello.
Yes, sir.
Yes, sir.
Okay. Fine. This is just to give idea. This is the cyclotron. Just we wanted to show how the cyclotron will look like. The particle accelerators. Okay. Having the different energies are there. Just I wanted to show the figure. Coming to grid, this is the device. One last type of the figure is given. That is known as generator. It is a device. What it does. What is the uses of this thing? Later slide we'll see. This is just I am just giving the diagram. Just giving the figure to get that. This is supplied by Brit. Brit indigenously manufacturing these generators. Now, coming to overall, if you see the thing, left hand side that my slide that the reactors are shown in the that is VRC Trombay, from where these things are produced, radioisotopes are produced. Then the right hand side, the automatic remote handling tubes are seen. There the processing is done because all are lead shielded glove box is there. Then nothing will come out. Only radiation also will not come out. Safely one can handle and process the radioisotopes, and purification also done. The third one is done in the quality control, where the quality control testings are done. Then after testing, it is given to the patient dose. The clinics it is given, and the patient is getting it. It's overall the contribution by DAEs for the societal benefit they are giving the thing. This is only because no other. If you are importing the all radiopharmaceutical, it will be so costly. The presence of BRIT or DAE is really controlling the thing, and we are able to get a lot of radiopharmaceuticals for the cure of therapeutic as well as diagnosis of cancer. We are able to use it. Coming to types of radiopharmaceuticals, there are many types of radiopharmaceuticals. This topic itself is a very big topic. Just I want to shorten it because our focus is to concentrate on the quality control. Now, types of radiopharmaceutical: ready-to-use radiopharmaceutical, generator-based radiopharmaceutical, positron emitting radiopharmaceutical. Now, these are the three types of radiopharmaceutical we are having it. Now, one by one, we will see. Ready-to-use radiopharmaceutical. Only one example. A lot of radiopharmaceuticals are there, but one example only I would like to consider because I want to again focus on our quality control. So, but at the same time, we also should know what type of radiopharmaceutical we are producing. We are producing many types of radiopharmaceuticals. All the three we are producing in bread. First one is ready-to-use radiopharmaceutical. That is simplest example I have taken. Sodium iodide. Sodium iodide is a normal chemical, you know, sodium iodide. That is used as a radiopharmaceutical. When it is labeled with 131, iodine 131 is the beta gamma emitter. That is when it is mixed with the, so when it is labeled with or mixed with sodium iodide, or we can prepare sodium iodide. What will happen? This will become a radiopharmaceutical. In addition to that, some of the additives will add it, so it will become a radiopharmaceutical. Its function. What is the function of this thing? It is again that all the function. I told you this. Most of the things are used for diagnosis of cancer or diagnosis of functional imaging. We can take it. Whereas in the case of X-rays and other things, we cannot detect the function that the static image we can take it, or the anatomical structure we can get the ideas about the using the X-rays. Whereas here, emission image we are taking. Whatever the radiations are emitting from sodium iodide 131, that emission image is captured by gamma camera, and that we get the functional imaging as well as static image. Everything we get about the organs. Coming to the treatment, the same radioisotope, we can use it for treatment also. In what way we can treat the thyroid cancer also, we can treat it. One can wonder how we can use the same thing for diagnosis, same thing for therapy. Only difference is for diagnosis, we have to take it to the inject into the humans very low activity. That is very low activity, microcurie level, very low. 1 millicurie is 10,000 times less is microcurie. So one microdegree, 50 micro, 25 to 50 microdegree level, if you are taking the thing, it will goes to thyroid. Then the function of thyroid can be diagnosed using this particular radioisotope, that is radiopharmaceutical, with low activity. Whereas if you take the higher activity, millicurie level, if you take 25 millicurie, 30 millicurie, or 10 millicurie, that depends on the size of the cancer, one can treat the cancer patient. This is for therapeutic purposes can be used. So only one isotope we can use for two purposes. The name as now the current name people are using for theranostic pairs. Theranostic pairs, here the pair is not there. One is sodium iodide, low activity used for diagnosis, whereas higher activity used for therapy. The theranostic is meaning is theranostic. One is talking about therapeutic use, another is used for diagnosis. Both the things are combined together. Theranostic. Now, importance of specific activity example. I told you what is specific activity. I explained. Radioactivity unit mass is called specific activity. In the case of the high specific activity, number of atoms will be radioactive atoms will be more. Why it is so important? Because in the case of again sodium iodide is injected into the body. It is taken up by specifically taken up by the thyroid. When it goes to thyroid, the competitive atoms, non-radioactive atoms and reactive atoms will compete. If the activity is low specific activity is there, then what happens? Low specific activity is there, then the radiation dose given to the thyroid will be insufficient. If the radioactive atoms are more, then the thyroid will get the proper dose. So it will be cured. Therapeutic purpose will be served. If the low specific activity is given, then the competition between radioactive atoms will be less, non-radioactive atoms will be more. Then naturally non-active radioactive atoms will occupy more. So then ultimately you will not get the proper dose. So hence the specific activity is very, very important in case of treatment. This is one example. Another example also I can cite in the case of peptide. Peptides specific tumors, they are all peptides are specific for the receptors. When they go to the receptor, if the low specific activities are there, receptor will be occupying with the low activity peptides. So naturally, the dose will be getting will be very low. As a result, the therapeutic use will not be much effective. Coming to the generator-based radiopharmaceuticals for diagnosis. So generator-based radiopharmaceuticals are manufacturing grade, that is molybdenum 99, technetium 99 generator. Why the two isotopes are mentioned? The technetium 99 is a medical isotope, whereas molybdenum 99 is not a medical isotope. What we want is molybdenum is the parent element. From that, when it is decaying into technetium 99 isotope, radioisotope. Like molybdenum 99 is having the half-life of 66 hours, okay, almost 2.5 days, more than 2 days. Whereas in the case of technetium 99 is having the 6 hours. So 6 hours is there. Then what we can do? Only we can sell it to near to the place we can supply to nearby places. If I want to give to some distant places like other than Mumbai, Pune, or some other part of Maharashtra, some other places we want to go, or some places we want to supply to the thing, it is not at all possible. But how it is possible? By making a generator, now it is possible. We have made it possible because molybdenum will be loaded in the generator. Then it will be transported to the different places. Then it can be eluted. In next slide, I will explain here that you will be able to understand. So this will be useful. But technetium 99M is the pure radioisotope we will get from the generator. Then we can formulate the radioform. But cold kits are also we supply. The cold kits are nothing but it is a radiopharmaceutical. That's not pharmaceutical. Pharmaceutical form we will give that cold kits. When you add technetium 99, because of this different oxidation state, it can chelate with different compound to form different radiopharmaceutical. That is a very, very ideal isotope. Then it has got the different oxidation states. So So we are also preparing different cold kits. One is for heart imaging, brain imaging, liver imaging, kidney imaging, different imaging purposes. We are making the different cold kits and we supply hospital. In the hospital, what they do, they cold kit, they take it, all are sterilized, all are called QC checked, all the things we give it to the thing, and the generator also QC checked. When we supply both the components, as and when required, they will elute the technetium 99, they will inject into the cold kit vials, and they will formulate the radiopharmaceutical, and according to the requirement of the thing, they will inject to the patients. This way, the radiopharmaceutical is used. I hope this is clear. Now, next diagram indicates this generator. In the generator, what are the things are containing? One is column. Outside is your lead shielding, because all radiation should be kept in the protective things. So it should be lead shield that is used. Lead is having the very high density, so it can cut off the rays. The rays will be coming, but we will be protected. The radiation will not pass through the lead. So it is a safety for the user as well as the supplier. That is those who are handling the thing to be useful for. Now, what is happening? The alumina is loaded in this. When parent element, molybdenum-19 radioactive things, it is when it is loaded. What happens? Alumina has the capacity to absorb only the molybdenum into the matrix, and rest are pure. When you are eluting with saline, the physiological saline solution, when you inject it, saline solution, we can collect the eluted while technetium, while pure technetium pertechnetate, we can collect it using the saline solution. Then what we get is the medical isotope we are getting. So molybdenum totally retained in the alumina column. This is the purpose. And the cross section is given this side. Coming to the why we need generator. Now, once you understand the generator principle, that means the daughter element is having the 6 hours of life. Okay, where medical isotope, that non-medical isotope, that molybdenum is having 66 hours of life. We have taken the advantage of molybdenum 66 hours. We load only molybdenum. Okay, molybdenum will decay to technetium 99. Okay, the technetium 99 will grow. Okay, one is reducing, one is growing. Then every 24 hours we can elute out technetium 99. Whereas if you are supplying 66 hours, like we cannot supply even to 2 different hospitals nearby. Also, it's very difficult to supply. In order logistic problem, to avoid the logistic problem, this generator was invented, and this was really useful. Find useful. Globally, people are using this technology generator. Generator also useful for the protection of radiopharmaceutical. Now, coming to positron emission chromatography. Now, throughout the country, you must have heard about cyclotrons. Cyclotrons are going on increasing because of the valuable contribution by F18 products, mainly of FDG, fluorodeoxyglucose. This you may be now familiar with the thing because of this COVID. Deoxyglucose has become very popular. The Reddy's lab and INMAS has produced the thing. So you may be DRDO has produced. So this you may be familiar with deoxyglucose. It's the thing that 2 fluoro in the second position, fluoro F18. That F is labeled in the position. That is F18 is incorporated in the second position. It becomes fluorodeoxyglucose. This molecule is called the millennium of the molecule because it is used in cardiology, neurology, even psychiatry is used, oncology, different fields it is used. Therefore, this particular molecule is known as millennium of the molecule. Molecule of the millennium, we call it FDG. Okay, now FDG is a radiopharmaceutical, specific radiotracer used in medical imaging, but only positron emission to PET tomography. That's best PET modality they use it, and mainly used for the diagnosis because of the half-life is very short, 110 minutes. And it is a positron emitter. What is positron emitter? Positron emitting radioisotope. Positron is nothing but beta with positive charge. That is electron with positive charge. It's coming out. That is having very high energy. So it is very much a PET technology. Why it is popular is because the resolution is very high. The sensitivity is also very high. That's why it has become more popular. Now coming to shelf life. shelf life or expiry date is very, very important for radiopharmaceutical. I told you the preparation depends on the primary physical half-life of the radioisotope and the radiochemical stability. What is radiochemical stability? We'll see. Radiochemically, it should be stable also. The isotope, suppose if you are taking simple example, I will take sodium iodide. Just now we have seen sodium iodide, because of the radiation, if it is decomposing to some other form, it is getting oxidized sodium iodide or sodium periodide is forming, then it is not radiochemically stable. That should be in the intact of sodium iodide only. In iodide form should be there, not in other forms like iodide or any other form. Therefore, that radiochemically stable, we check the radiochemical stability, then we will arrive at the expiry date. Expiry date, how it is fixed for radiopharmaceutical? First, physical half-life we will see, and stability of the radiochemical. If it is intact, we will periodically check before the introduction of the product into the market. So we have to submit the reports to the agencies. Then they will study. These are the radiochemical stability, how it is stable, all things they study. Then they will approve it. That regulatory aspects will come later. Now, many radiopharmaceuticals contain radioisotopes with very short half-life. Such people there are to have very short shelf life. Many, I told you, the 6 hours stability is there for the half-life is there for Section 99. Whereas F18 is having the 110 minutes for half-life. So the as per the half-life, this also will get reduced. Shelf life will be reduced. Certain formulation we have to use it within 2 hours, within 3 hours we have to use it. Now, importance of QC of radiopharmaceuticals. What are the things we are doing for radiopharmaceuticals? Purity, all the things, the attributes like this is almost common for the thing. Purity, efficacy, and safety, because of radiopharmaceuticals intended use for human use. Since it is used in the humans, these 3 factors, mainly purity, efficacy, and safety, are most important. Since we are intended for human use, extra precaution, extra care should be taken to the quality control. For stringent quality control measures, or even slight amount of the deviation is there, we create the batches. That is very, very important. Okay. And the case of QC also ensures the reliability, safety of the population, thus ensuring the quality product to the patient, not defective products. Always reliability is very, very important. Safety is also very important as far as the radiation is concerned, in the case of radiopharmaceutical. And it also ensures the safety of the regard to radiation is taken. How much radiation can be taken? How much should be injected? All the things are very much verified using the nuclear medicine physicians and health physicists and the technologists will all together, they will decide the thing, then they will come out with the product. So the safety point is also taken care. In the case of before injecting, they will see everything, how safe it is, animal study, everything is studied, then only they will come out with it. So the safety is also taken care by the QC analysis. Let us see what way we can. Coming to the QC of radiopharmaceuticals, what are special tests we are carrying out for for radiopharmaceuticals? We should know. In the case of radiopharmaceuticals, we carry out because of radioactive component is present, the special tests are carried out. If the radioactive component is not there, it will become pharmaceutical, same pharmaceutical. So it should be, second thing, difference is immediately it should be completed. All the tests should be completed immediately, and the test should be some of the things. We make 2 batches, morning one batch, evening one batch. That is one particular product, gallium-68 positron emitter product. We are in bread. We are now also carrying out in bread. Morning one batch will be produced, evening one will the operate that the hospital person will coming to the washi gate, and they will take it to the nearby hospitals. Some of the hospitals are requiring the particular product, and for the 1 hour, within 1 hour we have to make it. Within half an hour we have to complete the test, and we have to deliver the things. So all the things should be completed immediately. Evening also 1 more batch will be taken. Again half an hour we will have to complete all the tests. Simultaneously, all the people will be for 3 to 4 tests are immediately we have to test it, and we have to see the result, then it will be supplied. Then customarily what the thing is, except one test. Customly, the batch is released. This is very special to this radiopharmaceutical test. The batch is released before the sterility testing. Why? Because daily I told you, daily the batch has to supply to the patient. The radiopharmaceutical we inject to the patient. What we can do daily, we are making 2 batches. The 2 batches, the sterility test, all of you pharmaceutical people will be knowing, for sterility test, it takes 14 days to complete. Right? For 14 days, we cannot wait for the radiopharmaceutical. In this special case, the parametric release is done. Parametric release means without passing the test, we do the thing. What is the importance of parametric release? That we will see later. Why it can be complete, why it can be supplied like that, and we will see. But it is a global phenomenon. Throughout the world, this practice is continued. What is the reason for this reason? That also how that it should be done. Coming to, we are doing, there are 2 types of tests. One is physicochemical testing, another biological testing. This test is almost common: clarity, visual inspection, pH, radioactive concentration, radiochemical purity, radionuclide purity, particle size, chemical purity, residual solvents, catalysts, other impurities, starting material. This PCT for physicochemical test, I am not going to see the explain this. Mainly radioactive concentration, radiochemical purity, radionuclide purity. These 3 part is very, very important. The special tests I told you for radiopharmaceutical, these are the special tests we are going to see in this coming slides. And pH also important. In one case, I want to, classical example is there, good example I would like to give. Now, this chemical purity, the rest of things are similar to pharmaceutical. How we are doing EC spectrophotometry, whatever the analytical instruments are there, we can do. We can check all the other impurities, non-radioactive things. Whereas, biological, coming to biological testing, sterility test, bacterial introduction test, animal biodistribution. That is the in vivo test also done. So all the 3 tests we are going to see in addition to other tests. Total 6 tests we are going to focus on it. Now, in this case, we will see importance of pH. One classic example, why I have taken only the pH. pH in the case of particularly sodium iodide. Many of you knowing about the sodium iodide as such, it is a volatile, iodine is volatile in nature. Iodine is volatile in nature. If it decomposes into the iodine form, what will happen? It will be volatile, it will come into the air. Ordinary iodine comes into the air, is there no problem. But radioactive iodine 131 is gamma and the beta emitter will come, it is harmful, it will inhaling through the, will be inhaling and will be going to our body system, it will, radiation damage will be done. And it's very dirty isotope because of this volatile nature. How to handle how to avoid the volatilization is pH. When you supply to the hospitals, sodium iodide solution also we prepare it. Okay. Sodium iodide solution, what we do, we make it alkaline. Sodium hydroxide, particular molarity, not very high pH, very around 8 to 10 pH, it should be there. More than 8, it should be present. When it is there, then it will be intact as sodium iodide. Otherwise, in the acidic condition, it will be decomposing and will form the iodine. So iodine is, since iodine is volatile, so it is dangerous. So the pH is playing a very important role. In addition to stability, integrity is also maintained in the most of the pharmaceuticals. Because when you are injecting into the patients, the body system in vivo, the pH integrity should be maintained. Otherwise, it will go to the suppose myocardium that the heart imaging agent should go to kidney, will go to kidney imaging, some other organ it may go. If the intact is not there, integrity is not there, it will be going in a different way. Okay, therefore, in order to keep the integrity and stability of the product, pH has to be maintained. That's what the good example is sodium iodide. Now, what way we have to test this? Generally, people think that pH meter will be the accurate reading. But here, that what is very important is that accuracy also is required. But the pH paper, narrow band pH paper, is we are using it. That is almost almost like pH meter works. And why we are using the pH paper? Because daily we will be using the different radioisotopes. Daily, suppose 3 different isotopes are coming. One is F18, another is gallium-68, another is iodine-131, another is P32. All are different having different radioactivity and different that is emitters. One is gamma emitter, one is beta gamma emitter, one is positron emitter. So it will be getting contaminated. The pH, we should have so many pH meters, which is not advisable. So better to have one pH meter, and the pH paper should be validated against standard paper. Then only we should do the paper. That is always good. And coming to the, when we are doing the quality control test, this is the special thing we are taking, precautions we have to take. Since the products are radioactive in nature, all the tests should be carried out in few modes with good suction or negative pressure, we can say. What is the few modes, all of you know it, and with good suction. What is the good suction? Is around 0.5 meter to 0.8 meter per second. That much suction should be there. The air should be drawn continuously in the few modes. That arrangement is the suction is given to the each food. Why it is very important? Radioactive, and the fumes or the fume wood also should be adequately shielded using lead bricks. Lead, all of you know, lead is having high density, but the radiation will not come out of the lead, and it is protective. And in addition to the suction, we require lead shielding also. So with these two things only, we have to handle all the radioisotopes or the quality control. Everything should be processed or tested inside the fume. Why? Because of safety of the person who are handling the thing or who are testing the for radiopharmacy. What is the use of suction? I told you. Suction, why it is important? Suction means that the exhaust is there. When the exhaust is there, what will happen? That any volatile, mainly it is useful for the radioactive iodine. That is sodium iodine. The iodine is a volatile material. In order to avoid this, when in case of any escape of iodine, it will not come out of the sink, the person will not be affected. Therefore, the suction is very, very important for the safety. Second one, lead. The lead shielding is very, very important for the protection from the radiation. So, two things. One is volatile nature of the iodine. Another one is shielding from the radiation. These two precautions are taken. Then, another big problem is radioactive waste. In the case of solution, we are testing. After testing the thing, we cannot give to the supply. We have to throw it. We cannot, very small, of course, very small quantity, 1 or 2 ml only we take it. For different tests we have to carry out, we wasted. After the testing also, what then we will be collecting the different containers, and which are kept in the fume room, where the exhaust is there, always there. Then after a particular half-lives, say 10 half-lives, generally 10 half-lives, the activity will come to very low thing, then they will be discarded as per the monitoring health physics. We have got one department, health physics department is there. There the people will be knowing the dose calculation and how much radioactive decay. They will monitor how much it decayed, how it has come to the background level. They will check it, the pH, everything they will check it, then they will dispose to the waste management division, to the BRC. We have got waste management division in BRC. There, all the waste are discarded. Since they are all very short-lived, there is not much problem. And delayed decay, that is alarm as much low as possible is reduced as possible. We have to discard it. Delay, first thing we have to keep it for more time and decay. It will allow to decay and it will be coming to the normal pattern. Coming to the, when we are handling the thing, always the absorption sheets are used. Shoe covers, above the shoes, we are using the shoe covers also used. Why it is used? Because inside the laboratory, barrier is kept. When you go into the laboratory, you have to wear shoe covers, apron, and absorption sheet and TLD batches that we are. Next point is coming. Thermoluminescent dosimeter. All things we should wear, and working with the goggles also we should wear it because of radiation. Absorption sheet, in case of a contamination happens, the contamination will be affecting us. So the radiation will come out. So contamination monitor will monitor it, and the contamination there immediately we will have to throw. So periodical change is always a practice for precaution. Then coming to the dosimeter, thermoluminescent dosimeter batch also, each person will have the personal dosimeters. Always before entering to the lab, they have to wear it. This is not for protection. This is for monitoring. In case of accident. Suppose some flow is not there, exhaust is not there. Sometimes exhaust failure is there. Some people are testing it. That time iodine release is there. Then it will be deposited in the thermo. It will be recorded in the TLD batches. Then the person will get more dose. Then that will be. Then we will come to know this person is. Then they will be allowed to rest for some days. Then when it comes to the normal, then we will be operating again. So these are the precautions we have to take it for wearing this TLD batches. Then in the case of course, we have got enough protection is there. Radioactive air monitors, gamma monitors are so many monitors are in front of the food. We are placing 2 to 3 monitors are there in the whole area. Okay, around they find the square feet area we are getting area that is sufficient for monitoring air activity. When the volatile any radioactivity is coming in the air, immediately the beep sound will come. We'll come to know that then people should vacate the place for some time. After some time, it will go away because how it is going away. One more thing I would like to tell you their point here that not like other laboratory air condition is there, but what is the thing? 8 to 12 air changes per hour is taking place. Fresh air will be from 8 to 10, 12 air changes per hour. It will be on changing. That is another precaution we have to take. Then, coming to the instruments. So instruments part, just we will see the main instruments, dose calibrator. As we will see that TLC scanner, radio scanner means radioactivity scanner, HPG, high purity germanium detector, multi-channel analyzer. These are the 4 main instruments used in the use of radiopharmacy. I told you special tests. What are the special tests? These are the special tests using these instruments. We are going to know. So, important terms again: radioactivity, radiochemical purity, radionuclear. Radioactivity already we have seen it. Again, number of radioactive atoms undergoing nuclear transformation for during time, the spontaneous emission of radiation from the unstable nuclei. Okay, it is expressed in curie, millicurie, becquerel. Becquerel is the SI unit. 1 becquerel is 1 DPS. 1 curie is 3.7 DPS. These are the units of radioactivity. That is quantity. How much quantity we have to give to the patient? That quantity we are measuring using dose calibrator. The picture of dose calibrator. Where the cylindrical one we are showing is, in the cylindrical one, the radioactive vials are we are keeping it inside, and the left hand side the monitor is there. There different buttons are there. Depends on the different radioisotope, different buttons it is already calibrated. If you switch on the particular switch, in technician 19, if you want to measure it, it will be seen in the display, and we can measure the radioactivity. And we can take it out. The isotope dose calibrator is very, very important. Radioactive concentration finding out is very, very important. As per the regulation, what is the thing that isotope calibrator has to be calibrated proper way as per the regulation. That is constancy. Daily we have to check the constancy, accuracy annually or after required, linearity quarterly or after required, geometry after required. These are the tests are very, very important. Why these are the constancy daily, the radioactivity counts are measured daily, particular activity is measured, and same while we have to take it and daily we have to see as per the decay. Suppose 1 millicurie is there, after 1 half-life it should show 0.5 millicurie. Again the constancy, again the 0.5 millicurie to 0.25 millicurie. That way we have to take say 100 millicurie will become after 1 half-life period 50 millicurie. That constancy we check daily. As per the decay factor, we can calculate how much activity should come. Constancy is done. Then accuracy annually or after required, we will do it accuracy. For the 10 millicurie, that is nearer to accuracy is nothing but near to the true value. If we expected value 10 millicurie, you should give at 10.1 or plus or minus 1. 10% if it shows it is okay, or plus or minus 5% is also because 10% is accepted in this case. Linearity is also it should give the linearity. That's also there. That also we'll do it. And geometry. When we come to the geometry, what we do different vials, 1 ml vial, 2 ml vial, different vials we take the radioactivity and different geometry it should give the same activity. That way we the linearity we measure 5 is we measured from microcurie, very low activity, very high activity. Linearly should be that 1 microcurie, it should give 1 microcurie. Then we see through 1 millicurie, then 10 millicurie. Then like that we go on increasing. The linearity should also will follow. It should give the proper reading. So all the tests are very, very important. Why it is so important? The importance of radioactivity, once it is the dose calibrate, very important testing instrument in this case, because the radioactive concentration is more. In the case of if it is so, we have to supply 10 millicurie. By mistake, we have the dose calibrator is not working. We are thinking that 10 millicurie, but it is showing 15 to 20 millicurie. What will happen to the patient? And the first thing, the patient will get extra dose, which is unnecessary dose, and which will damage the other organ or it will damage the other cells. Secondly, the operator who is going to inject or going to handle the things, the operator as well as the nuclear medicine physician will also get more dose because of extra unnecessary dose. Because they will be wearing the proper protection for this thing, and they will think that this protection is sufficient. But since extra activity is injected, is taken, so unnecessarily dose, they will also get the dose. If the radioactive, then coming to the this quality image, what will happen to the image? We like to say in diagnosis, if the radioactivity is less, the quality of the image will be affected. At the same time, the radioactivity is less. In the case of therapeutic case, what will happen? The proper therapy will not be given, and radiation will not be proper, so that it will be therapy will not be successful. And again, we have to repeat it. So, in the case of more also it's a problem, less also it's a problem. So, therefore, the radioactive concentration measurement is very, very important. Therefore, we use the different tests, always test and calibrate. Then coming to another important property. Hello, is it okay? Hello.
Yes, sir.
Hello.
Yes, sir.
Okay.
Yes, sir.
Okay. Dr. Patel, no? Okay.
Yes, sir.
Okay. Coming to radiochemical purity, it is tested using one of the methods mentioned. This is the general chromatographic methods. One is paper chromatography, other is the thin layer chromatography, HPLC, high performance liquid chromatography, electrophoresis, followed by any one of the methods. We are standardizing it and we are measuring it. When generally used method is paper chromatography, thin layer chromatography, convenience sake and fast. HPLC is also now most of the things in grid we have standardized for HPLC because instrumental methods are always preferred to. Here the manipulation is possible. In the case of paper chromatography, thin layer chromatography, manipulation is possible, whereas in the case of HPLC manipulation is not possible because we have got the program and all the things. Even the injection is done, the time will be given. How much time? Second injection, all the times will come. So with the certain program, we can control the manipulation is not possible in the case of HPLC. Therefore, people prefer HPLC method. Even though we are also using paper chromatography, thin layer chromatography, and all the chromatography, some of the things I am seeing the thing, radiochemical purity checked by paper chromatography, thin layer chromatography. It's not like that. All the things should be followed for radioactivity. Detection will give you radioactivity purity. If you don't, only paper chromatography method, how will you find out? No, it's not possible. You have to scan for the radioactivity or detect for the radioactivity. Then HPLC should have HPLC. When you have the HPLC, it also should have the radioactive detector. We have got the radioactive detector connected to it. So when you pass through the thing, it will pass through the radioactivity. Then it will be determined. The radiochemical purity will be determined. Then this is a radio chromatogram scanner or radioactivity scanner. We call it. The top one in the cylindrical vessel is nothing but the detector. The plant, either the instruments are there, two types of instruments are there. The platform, either the platform is moving or the detector is moving. In our case, the detector is moving. The plate is PLC plate is kept. When the radioactivity, then right from the beginning, we can start it. And when you start it, what happens? The graph we are getting like this. The RF is 0.5. The radioactivity beam peak is we are getting like this. And some of the start as the solvent front also some small peaks are coming. That is slightly above the background. Background also we can fix it. So from this we will come to know our the product is pure. Then how we are computing? Suppose in case of we have got the scanner. In case the scanner is not there, what we do? This is where we can do it. The TLC thing, we are developing the TLC plate like this. At the middle of the spot, we are seeing the spot in the middle. RFL is 0.5. Then what we do? We cut the spot each centimeter from the middle of that area. 1 centimeter. 10. Suppose let us say 10 centimeter we are cutting. All the total counts we will cut and count in the counter. Okay, bell counter is there. When the radioactive counts can be taken, each centimeter the counts is taken. Total count, let us assume 10,000. That is counts per minute or counts per second. CPS or DPS will, CPM will come. The counts rate is the total counts are recorded. We can record it 10,000. Total counts we have to count it 10,000. And the RF value 0.5 is matching with the RF value. The spot is cannot be seen in the thing, but using the proper reagent, we can come to know what type of the compound we can come to the coloring reagent, we can do it. Because radioactive component will be very, very small. It cannot be seen. Like the spot will not be seen for UV or any other thing. But our coloring reagents are there. We may make the coloring reagent in the non-radioactive spot. Simultaneously, we will be adding the co-chromatography, the non-radioactive component also we will be putting it. Then it will give the color. That particular RF value is 0.5. That corresponding to counts also we will measure it. 9,800 counts will be there. Then the radiochemical purity is 98%. We compute like this. So in the case of scanner is not working or scanner is not there, another way of doing is cutting and counting. Yeah. This way also one can find out the RCP, radiochemical purity. This is very, very important. I think radiochemical purity. Definition. Definition we'll see later. In this case, these are the different things I have given. One is HPLC is given at the bottom. Paper chromatography is given at the bottom. How it is coming, the graph is coming. Then size exclusion chromatography also for proteins analysis. Just example. The HPLC thing you can see. Current testing of RCP. Tested using ultra performance chromatography. UPLC. You must have come across UPLC. Many of you may be doing this. With radioactive detector, one can have RCP can be determined. What is the advantage of using RCP? So now, many part of the world, in the foreign countries, many people have started using RCP. Now, our Brit is also taking up the UPLC with radioactive detector. What is the advantage? The green analytical chemistry. See, for analysis of even 10 analysis can be carried out in 100 ml of solvent. Whereas the HPLC requires liters of solvent we require. The solvent usage is very high here. Solvent usage is very less because the particle size of the thing is very low. So the theoretical plates are more. Resolution is very high. And all things are analysis can be performed completed within 5 minutes. It is very apt for the short especially for the positron emitters. It's very radiopharmaceutical, very useful. So UPLC. What we can do is what I can suggest you for those who are faculty members, UPLC method, you can develop it and that collaborating with Brit, all the tests, whatever we are carrying out the tests in the HPLC, one can convert it to UPLC, one can publish it. Those who are in the analytical line, analytical development thing, one can take up the PhD because I have seen some of the theses. UPLC method, only UPLC method for pharmaceutical analysis, one fellow has done it. Some other industry, the Saurashtra University. Similarly, one can take up the project also. Even the PRNS project, all the projects can be taken by the faculty members for developing UPLC method for radiopharmaceutical. Once you are linking with the radiopharmaceutical, we are Board of Research and Nuclear Sciences will give that one leading institute is there. PRNS thing is there. There they will give the funding. So using this thing, this I was long back I was thinking, but it's not materialized. When I retired, the 20 up to 20 it was not materialized. Now young you can think about it. UPLC, you can get the UPLC for your college, your institute also you can get it free of charge. That is BRNs will provide the thing, and at same time radioactive development you can develop at the BRIT. You can develop this particular method of analysis for all radiopharmacy one can do. Now radiochemical purity, why it is coming? We have to know once you know the thing radiochemical impurity, how it is coming. It is coming mainly because of improper purification. Improper purification, then the purification methods are not good. So naturally that impurities will come. Then another one, this one is radiolysis. It's the inherent property is radiation. Wherever radiation is coming, the decomposition due to radiation takes place. This phenomenon is known as radiolysis. So all the radiolysis, irrespective of whether or any condition you store it, radiolysis, some amount of radiolysis will take place. If the solutions or if it is stored in the solution form, much more radiolysis will take place because of the presence of, suppose aqueous solutions are there, water will decompose, and secondary free radicals will be forming, free radicals again will be decomposing things. So radiolysis will be more in the case of secondary decomposition. The third one is storage condition. Some people will not they will not be storing in a proper way. They will expose to necessarily light and the high temperature and pH variation is there. Then all things will be getting decomposed. They will affect the radiochemical purity. Radiochemical impurity, the presence, what will happen? Suppose the radiochemical purity generally we require 95% purity is required. If it is less, what will happen? First, it will affect. We are generally using for the diagnosis for the image. The quality of the image will be not proper, leading to difficult interpretation. The nuclear physician also will get difficult for the interpretation. They feel it. Then, in the case of therapeutic, what will happen? It will not be effective. The impurity is present. If below the specific is not met or the impurities are more, it will not be effective. Due to the impurity, that unintended target also will result in the organ damage. It also will affect the efficiency. You can get impurity. The iodine is containing some impurity. Instead of labeling that with iodine one compound, you want to labeling with iodine. The labeling will not be proper. That all things will be affected. In addition to biodistribution. Biodistribution, one test we are doing it, that also will get affected. Now coming to radionuclidic purity, it is defined as previously I have not explained the radiochemical purity. I think that slide is missing. I don't know. The radiochemical purity is defined as a fraction of radiochemical purity. This radionuclidic purity, but I will explain radiochemical purity is nothing but the fraction of the total radioactivity in the form of desired chemical form. In the case of radiochemical purity, what is the exact thing is sodium iodide should be present as sodium iodide, not as sodium iodide. NaIO3 should not be there. That is the chemical form. That is I am talking about. Please don't get confused because the slide is missing or I don't know. I'll have to see. Radiochemical purity, the chemical should be in the desired form. The radioactivity should be present in the desired chemical form. Whereas radionuclidic purity and radioisotope purity should be the fraction of total radioactivity in the form of desired radionuclide or radioisotope. Example is in the case of sodium iodide 131, we want to formulate sodium iodide 131. But iodine should be present in the form of iodine 131, not containing any other isotope other than iodine 131. Not even 125 should not be present. Not even any other radioisotope. Technetium should not be present. Molybdenum 99 should not be present. Any other radioisotope should not be present. So these are the very important radionuclidic purity concept. So using how we are testing it, using high purity germanium detector, we are having it. The detector is we are having. Using that detector, we are finding out radionuclidic impurity. Then how it is coming? We also should know the sources. Radionuclidic purity, because this is all the things are coming because of production process. The production process are not proper, then it will lead to purification. There all the production also, the purification, other things are there done. There the chances of contamination using the same vessel for some other radioisotope vessels are used, then the impurity may come. And again, that depends on it depends on the target material. What we do in the target material, the material we want to convert into radioactive form. The target material we are keeping in the reactor, we are bombarding with the neutrons, or we are in the case of cyclotron, we are accelerating the particle and bombarding the target material. So it will be converted into radioactive element. Okay, when we are converting the radioactive element, the target material should how pure it is. That depends on enrichment of the enrichment is see certain things we cannot improve. Certain target material purity cannot be reached. Purity is very difficult. So people will use the target material. Due to that, radionuclidic purity will come. So most of the things are pure form. If you use it, then radionuclidic purity will be very high. Then again, what is the effect of this thing is impurity leads to more dose to the organ. Again, some of the impurities like alpha emitters coming definitely will be very harmful, and beta, gamma emitters, then again it will not only affect the thing, it degrades the radiopharmaceutical, it affects in the case of diagnosis, it affects the quality of the image. So what is the purpose of diagnosis? Using the gamma camera, we have to take the picture or image is taken. The image quality is not good in the radionuclidic because so many impurities will come and mask the background will not be found. Radionuclidity arises during the production depends on the enrichment. This we already seen. Then this is detected using high purity germanium detector or multi-channelizer. These are the HPG detector we are having in our laboratory. This is one useful, and the multi-channel is also useful for them. That is overall multi-channelizer, and this one is nothing but the gamma spectrometry. Using the gamma spectrometry, we are taking the gamma spectrum, so it is analyzed. That each has got the particular energy will be shown. Then we will come to know the energy of the gamma rays. Then we will come to know this particular isotope is pure. No other peak is coming without this. Now coming to that's all about the radiochemical purity, radionuclidic purity, and radioactive concentration. Coming to the biological, the PCTs, these are the main thing: radioactive concentration, radiochemical purity, then radionuclidic purity. These three are the special tests we are doing for radiopharmaceuticals. In addition to that, this is a normal test. All of you know that bacterial endotoxin is liver. It's a lipopolysaccharide found in cell wall of gram-active bacteria. This is a typical type of pyrogen. If small amount is present, it also will lead to even to lethal. It may be lethal. It is a particular limit in some cases. Okay, so it should be within the limits. So here, the biological tests are very, very important for all parameters. Wherever injectables are there, that we have to do the BED test, DELITE test, and the animal biodistribution. In case of radiopharmacy, we are doing it. Then endotoxin should be within the limits. In the case of limit is 25 endotoxin unit per ml and should not be more than 175 EU per volume. The volume B, the volume injected. Why 135? Generally, 25 into 7 is multiplied by 7 ml. Generally, we are injecting. Therefore, the 175 limit is fixed. EU per volume is where V is the volume. Volume is 7 ml. We are injecting. In the case of radiopharmacy, therefore, the endotoxin limit for the radiopharmacy is 135 EU per 7 ml. Coming to bacterial endotoxin, how we are detecting it? The test is very well known to the pharmacists because they are also doing the same test similar to this. So we are also doing the thing. Why I want to give this test here because one more new instrumental based test as well. That's what I want to highlight it. The principle is same. Lysate, the LAL, law reagent, lysate limulus amiboside reagent is when it is reacting with that pyrogen of gel clot is forming. Gel clot is forming, then it is the presence of your endotoxin. So this is done at 37 degrees for 1 hour, almost 1 hour test is done. Now, recently, Charles River, one company, American company, has introduced this thing. It is FDA approved system. Using portable test system, Windows safe analytical instrument, using the same principle. But in the spectrophotometer based, it's a color chromogenic agents are inside it is there. When it is reacting with the thing, it will form the coloration. Some colored compound is formed, then optical density is taken, absorption is taken, and pass or fail results will come. Okay, it is mainly useful for the F18 based radio purposes because they are having 110 minutes off-life. So it has to be finished within 1 hour. So all the agents are able to complete the thing within 25 minutes to 20 to 25 minutes. This is the PTS endosafe system. A very small portable system. Anybody can take it out and do the analysis. And here the cartridges come. This is the cartridge-based system. What is happening? The cartridges are there. The 4 rounds they are showing. Intertoxin spike and sample reservoirs are given in the thing. There, the very little amount of 550 microliter of the sample are injected. When it is injected, it will drawn inside using the pump. There, the pump connection. Then all the things 4 will be reacting with the reagents and forming the particular density, optical density, and it will be measured by the spectrophotometer instrument and give the results within 20 minutes. It's a wonderful thing to have especially for radiopharmaceutical. Only one thing is, in the case of radiopharmaceutical, what is the advantage is, it is very low quantity of the thing injected, it is safe, and once it is injected, what happens, the radioactive waste generated is less, the cartridge can be packed and disposed very carefully, without much contamination. So, in case of radiopharmaceutical, very useful. And coming to sterility, it is similar to pharmaceutical. Any test bacteria and fungi are tested using 2 different mediums. All of you know it. It is the 14 days test. The clear solution indicates the product is sterile. Okay, why it is essential? You know, the blood is sterile in nature. So when the things are injected, the sterility should be checked. Otherwise, it will lead to problems. Even to very strong cases, BET and sterility tests are very much important in the case of your injectables or parentals, we call it as. So it should be sterile there, leading to death also. Now, coming to animal biodistribution. Animal biodistribution is very, very important. Each batch of, now we are making 3 different batches in radiopharmaceuticals. Okay, one is heart imaging agent, another is liver imaging, another is the kidney imaging. All the things when we are making it, all the 3 will be sent to the animal house. We are having the British having the animal house. There, the BRC is having the animal. We send the product, and each radiopharmaceutical preparation after formulation immediately will be injected into the animals. And particular time point, the organ is this rat. It is done in the rat, Vistar rats or Swiss mice. It is given in the Monaco. We follow it. So when we are doing the dissection, which organ it goes, non-target and target, all the things we analyze it, and we give the result pass. All the things are done. Each test is done. Each pharmaceutical tested in 3 animals. Second, radioactive target non-target is determined, and the results are there. So, but the main problem is when we compare with the radiopharmaceuticals, in this case, every batch has to be tested for daily. The preparation is same product will be doing 2 batches. Even the 2 batches, the morning 1 batch, suppose we are doing animal biodistribution, we are doing, except for your ready to use radiopharmaceuticals and for the F18 and the gallium products, we are not doing it because it is not possible to do the biodistribution. During the initial studies, biodistribution is done. Okay, similar to pharmaceutical products are also not for biodistribution every batch. Initial time when they develop the product, they do it. Afterward, they do it. Only the technetium-based, the generator-based pharmaceutical, especially technetium-based radiopharmaceutical, we are doing this biodistribution. And now, one more project we can think about for the BRNS project is, the faculty members think about it. Biodistribution now imaging system has come. Rat or mice can be small imaging system. Even BRC has procured now currently. Okay, and what we we can do the analysis because this biodistribution is non-destructive method. Once you are injecting, after taking the image, we can reuse the animals also. That type of things are possible. Number of advantages, number of animals sacrificed, because we need not sacrifice so many animals per day. We are sacrificing minimum 9 animals per day. So much animals will be sacrificed. So much breeding is to be done. To avoid all the things, we can use the biodistribution. This method can be utilized. Imaging can be done, and one can take up the with along with bread pharmaceutical imaging. One can do what is to be done is imaging is a. This already I told you. What we can do. We have to do the comparative studies. Once you are taking the thing that radioactive injection we are doing, destruction we are doing, target how much quantity, quantification we have to do in the case of imaging. Imaging also slightly cumbersome. The calculation part we have to do when your imaging is done, how much quantity retains the particular organ. If it is correlated with the other method, older method and dissection method, this one is matching for consecutive around 12 batches, suppose 6 batches. So many batches you have to ensure this is perfectly matching with the thing. Then one can publish and bring it to the regulators, and they will also take it up with this. Yes, look, so far the quality imaging is not taken as given by any pharmacopoeia commission. So this can be also a good project for the pharmacy people who can take up link with your radiopharmaceutical. Okay. So you can learn how that research is done. You can do the imaging in their premises and do the correlation study. One you can publish also. This is a very interesting PRS project can be done. Now, parametric release. This is especially for the radiopharmaceuticals. Parametric release is mainly for the sterility test. Radiopharmaceuticals release prior to the completion of sterility test, because I told you it takes 14 days. But morning one batch, evening one batch, you want to give it to the patients. How will you supply to the hospitals? So sterility test. Without doing the sterility test, we are doing it. How we are doing it? Before that, globally what they do, we set up the laboratory and we test the first production facility. When you set up the thing, mainly for the short-lived radiopharmaceuticals like gallium-68 and F18 products, they do continuous so many batches they do, and they analyze the thing. They put in the sterility test record, they keep it. Then from the experience, how they do it, how the aseptic condition they maintain, their experience and history of the production. They do the production, how they maintain it, and they check the quality. Each batch is checked and perfectly after maintaining thing. And also we are following the good manufacturing practices also. Combining the 3 factors, experience and history of the production, all the things will come to know that our production is foolproof. Then we can ensure the parametric release. This is how globally this parametric release only for sterility test. Without doing the test, people are even the slight amount risk is there. The risk is nullified because of the experience, history of production, and trend analysis. Of course, one can do the trend analysis also. Can do it. From this, we'll come to know how our pharmaceuticals are superior or not. Now, coming to regulatory aspects. When we are talking about regulatory aspects, very, very important review, because radiopharmacy experience, initially the regulator was only 2 agencies were involved. One is Radiopharmaceutical Committee, another is Atomic Energy Regulatory Board. Only 2 people were involved. Okay. Now, 2 more people have come into the picture. That is Indian Pharmacopoeia Commission and Drug Controller of India. Of course, Drug Controller and Pharmacopoeia member FDA people will be a part of Radiopharmaceutical Committee, but they will not be controlling overall, controlled by initially, not now. Around 5 years back, up to 5 years back, say around 2015 onwards, the Indian Pharmacopoeia has entered into radiopharmaceutical domain also. Because we also want to have that IP in collaboration with, of course, with the BRIT members. They are doing it. You may be knowing Dr. Sivaprasad. Sivaprasad is a member in the IPC. So he is also helping to formulate that particular monographs. or what are the defects, all the things, educating the IPC members again. Okay, now radiopharmacy committee is how will be the thing one by one. Radiopharmacy committee is an internal committee. It is constituted by chairman of atomic energy. Chairman of atomic energy is formulating this committee. This committee will be consist of different pharmacists, radio chemists, then production people from radiopharmaceutical, and production the nuclear reactor people, then your FDA people, pharmaco IP members, all the members and nuclear medicine physician, all will be the members in addition to QC members. Coming to the role of radiopharmaceuticals. What is the radiopharmaceutical committee? Approve whatever we have got monographs for production as well as the QC. All the approvals will be given by radiopharmaceuticals. Approval of change, deletion, and improvement in the procedure of QC on any modification in the production procedure. So get the approval from radiopharmaceutical committee. This is up to 2015. This committee was so strong, and whatever the total over was with radiopharmaceuticals, they decide everything in this case of approvals, all modification, any change in that, they will approve. Now, this is you should know that this particular thing is RTC committee is applicable only for DA units, Department of Atomic Energy. Then we are producing, the British producing. Of course, in some places like Kalpakam in the south also they produce, trying to produce the thing. They will have the control. And in addition, RMC and radiological laboratory and BRC, three to four centers are making radiopharmaceuticals. They will be controlled by this RTC until 2015. Now, suppose somebody wants to outside the country, that is outside BRC, non-DAE people, some private people, some people have retired from Brit. They have started pharmaceutical production unit at Gurgaon. That is SDS Life Sciences. They have also started thing. There they are not controlled by the RTC committee. Who is involved in that? Drug control of India is the control. The DCGA is directing. So what they manufacture, the generator, that will be tested by Brit. Then it will be approved by DCGA. Then they can sell it. This type of arrangement is one. Role of Indian Pharmacopoeia Commission initiated the inclusion of monographs. So Brit radiopharmacy in collaboration with Brit. I told you that Brit, some of the members, senior members like Dr. Shiva Prasad, who is associated with your Bombay College of Pharmacy, is also very much involved in educating Indian Pharmacopoeia, and he is involved in the monograph commission. The role of Indian Pharmacopoeia, customarily the monographs contained within the national pharmacopoeia are the official source for pharmaceutical products. Any pharmaceutical products, we will have pharmacopoeial things only we follow. It is only the guideline we are supposed to follow. So that way they give the reference material they give. That's the role of. The role of AERB, Atomic Energy Regulatory Board. This is the independent unit of DAE. What they control is wherever the radiation is there. Don't anywhere. Let anybody start this. The radiation safety is concerned. That is very, very important. That is even beyond the DAE units. Also, somebody is starting newly. They have to abide by AERB rules. So they also have the control of the radiation safety of the production, QC facilities, packing and transportation, and radiopharmaceuticals. Here, the things what is very important, mainly that involve the radiation safety production. QC the additional, they come and visit our Brit facilities. For production facilities, they check the adequate ventilation, adequate shielding, everything they check. That is safety part. Any radiological radiation leakage should not be the all things they test. They come and inspect. QC facilities and packing. Why packing has been tested? Yes, special thing is in the radiopharmaceutical packing. As far as packing is concerned, vial will be again packed in the secondary container called that lead containers, because all the things are radiation based. So radiation will come out. To avoid the radiation coming out, we are having the particular thickness of that depends on the radioactive particular thickness of lead part is taken, will be packed inside. Then again will be taken in the thermocol again will be packed in the another container because surface dose should not come. That type of thinking, they'll be checking it. And transportation also, we have got the special transport for transporting the radiopharmaceutical to airport. That this also will have the shielding, lead shielding, and all the containers will be properly kept. The placement will be there for keeping the particular radiopharmaceutical, because in case of accident, it will not jump, it will not fall down. So that type of arrangement is there. With that vehicles only, transportation can be done. So all the things are radiation safety taken care by. Yeah, in addition to not only for the bread, but for the whole anywhere people are starting it, they have to abide by their rules. With this, almost we are coming to them. So I was telling the QC why when we do the QC, what are the advantages? With this, I would like to stop my presentation. Advantage of QC, we should know why QC is done. Ultimately, we know that the QC is done for the quality, to meet the specification of the product. Okay, the product should have the particular specification. If it is meeting it, it is well and good. The product. So consistency of the product. When the group is producing the particular product, we'll come to know that production group is functional, that is working in a proper way. Consistency is maintained or not. Because sometimes what happens, the consistency may not be good because of change of raw material. Sometimes the raw material may not be available from some particular companies. Some company we will change it. Then we'll have to check the QC. That time some variation is there. Again, we can tell that this particular product you are using this particular raw material. Then you change the product, you change the raw material, so that in order to maintain the consistency, the QC is having the real role. Then attract new customers. In what way the new customers are there? Because we have got Brit has got one very good project or one US project we got it because we were very much qualified for the thing. At the same time, we have got some HPLC that is instrumental based instrument. Most of the things are sophisticated instruments we were having. So that when we visit the US people visited, then they told that radioactive for the pharmaceutical, they went to different countries. Finally, they came to India. Then we have taken the project. The Dr. Suprasad was the thing that the Peregrine project was there. So because of this availability of new instruments and the sophisticated instrument and expertise, so they got attracted towards our thing. Production facility also we have got it, and the QC also we are well equipped. So based on the instruments, they got attracted, and we showed the capability, and 40 patient studies we have done it for monoclonal antibody labeling. We have done it at the contracts we got it. So like this, when they visit, some people visit your place, when you have got the good instrument with consistency, with a well calibrated instrument, definitely they will think, oh, but these people are doing a very good job. Naturally, that they will have that good impression. Then next one is the product quality. In the case of failure, in the water case, failure is there. Suppose the pH is coming instead of 8, 7.9, it comes, let us say. Then product quality we have to improve. We have to tell the thing that you people have to use proper alkaline or this condition to improve the quality. That feedback can also be. Then to understand the history and using the trend analysis. Once you know the history, I told you the history should be known for certain tests like sterility test. Sterility test, without knowing the test, we have been doing it. The trend analysis, the history, we can understand using the trend, we can understand the history, so one can maintain the quality. So these are the advantages or uses of QC in the case of mainly for the radiopharmaceutical area. With this, I end my presentation, and thank you very much for your patient listening. Any other question is there? Welcome. I hope all of you are clearly heard, or I don't know. I'm not getting the feedback. Tell me. You heard it clearly?
Hello, sir. Yeah, we really, yeah. Thank you very much for such enlightening session, and I'm sure our participants must have found it very informative. Before we take any questions from the participants, let me just sum up this session. Here, Dr. Padmanabhan, he has discussed for various types of radioisotopes. Then he briefly explained the basics of radioactivity, also discussed on the production of isotopes, and explained the flowchart of diagnostic applications of radiopharmaceuticals. He had given brief information on the generation of radioisotopes, given various examples of radiopharmaceuticals. In his talk, he also emphasized on the quality and the safety of radiopharmaceutical products. He further highlighted the special test, that is estimation of your radioactive component, purity, then radionuclidic impurities, which are time bound, and further elaborated on precautionary measures while testing the radiopharmaceuticals. He also showed the instruments which are used in quality control testing of radiopharmaceuticals. He had touched upon the calculations, and it's a very wonderful session, sir. With your permission, can we take some questions from the chat box?
You can take.
Yeah. So we have a question here first by Professor Krishna Priya. She is asking, usually for biological studies, the sample size is 6 or more. Why the biodistribution sample size is only 3?
That is
radiopharmaceuticals.
In the case of radiopharmaceuticals, 3 animals we take it. That is by the monograph. We follow the pharmacopoeia. You see, we take from other pharmacopoeia and we study. We establish and we study the things and we present it to RBC committee. With the approval of a monograph only, we follow it. We are not like only 3 is sufficient. 3 are out of 3. If 2 passes, then it is passing. 3 passes is well and good. 2 passes also it is good.
Okay, sir. Thank you. Yeah. There is a next question by Surya Goel. He is asking, sir, as you told, many radioactive therapeutic isotopes exhibit longer half-life. What kind of difficulties may arise while dumping the waste of such radiotherapeutics?
Yeah, very good. Now, as far as the radiopharmaceutical concerned, not very long half-life. Only one is urea C14 capsules we are producing it. That is for detection of H. pylori. Ulcer, to find out ulcer, H. pylori test we are doing it. That is known as breath test. When you take the 1 microcurie of this one, in the biological system, it will be eliminated within 3 days. It will be eliminated. Okay. What the thing is, if the H. pylori is present, it will be degraded to carbon dioxide. Okay. The urea is ammonia and carbon dioxide. It will be released immediately from the system. Okay. Even though it is having the physical half-life is 5,730 years, it will not be remaining in the body. It will be excreted in the body. The biological half-life is very less. So in that way, it is safe. But most of the radiopharmaceuticals are short-lived. It will not have any deleterious effect on the body because it will be well established before taking into the thing. All things are highly standardized. Throughout globally, these methods are followed. And C13, C14 urea, because of some difficulty in developing the C13 urea capsule, we ended up with C14 urea. Okay, C13 also, that is non-radioactive. That is very difficult to convert some. Whereas the detection of this one is breath test, easily you can detect it using scintillation counter. Therefore, we have taken up only one product, that C14 urea. That is 1 microcurie, very very low activity. We are doing it. Except that no other radiopharmaceutical is having the very long half-life, maximum 8 days. This one is the days only. Tell me.
Thank you, sir. Now,
not much.
Yeah, Dr. Sebi is asking, can you highlight the points to be followed while taking the PET scan of rat brain for Alzheimer's studies?
PET scan, see what are the precautions to be taken. This is only nuclear physician only can tell about this thing, exact thing. Okay, for this quality control point of view, I can tell what are the tests to be carried out for the FDG. Mainly FDG is only used for the PET scan mainly. Okay, for the thing quality control aspect, we have to the radiochemical purity, radionuclidic purity, and all the things should be tested. Radioactivity should be mainly tested. And in addition to that, the preparation of FDG, they use a different solvent and catalyst. All things should be tested for this. Special test should be carried out. That's all. Only RCP, RNP, radionuclide purity, and radioactivity. These are the main things we have to take it for as far as quality is concerned. Other things, precaution to be taken, or what is to be a condition for Alzheimer's disease or whatever the disease, that you have to contact only the nuclear physician. I am not aware of that. Okay.
Okay, here one question from Dr. Vaishali Shirsat. She is asking, in drug metabolism studies, which different isotopes are used to understand biodistribution?
Very good question. For the biodistribution, the technetium labeled pharmaceuticals are used. Sometimes iodine 131 is also used. These two radioisotopes are preferred for understanding the biological metabolism. Because animal thing, we are using a lot of experience in the animal biodistribution. The technetium 99 will be preferable. Okay, different types.
Sir, I have one question. You were discussing about the formulation containing iodine as a radioisotope, where you need to have a pH towards alkaline side so that you can reduce the volatility of the.
Yes.
Correct. Kind of all the other formulations containing other isotopes. Like
No. So, technetium isotope. No, this is one classic example. I told that pH of importance of pH in this case. All the things of pH is very important. Blood, the blood pH can take up the buffer activity is high. One can take it up, but because of the radiochemical instability of the iodine, we have the volatile nature of the thing. We have to take the particular pH in the alkaline condition. Otherwise, the range is some 4 to 8 will be the ideal things people are using it. Okay, different pH range using it. That highly stable. Okay, it is not alkaline condition we have to maintain for all radioisotopes.
Okay. Dear participants, if you cannot type anything in your chat box, you can raise your hand and the mic will be given to you. You can ask. Sir, I would like to ask the question about now suppose any other pharmaceutical firm, if they want to start production of such radionuclide, right now it is with GRC.
Now you can start. No, hello.
Yeah.
Now we can start. Now I told you that in Gurgaon, one person, ex-Brit, after retirement, he has started one company. He is working in that company. He only initiated the thing called SDF's Life Sciences. They are making generators, technetium-based generators, weekly around 10 to 15, 20, around 20 generators per week. It's a weekly production. Okay, weekly production they are supplying to the different hospitals. They are supplying in addition. Previously, only Brit was maintaining. You can also start. Only thing is radiation safety should be taken by the care that the ARB you have to approach, and certain things you have to submit the things, plan approval, site approval, certain formalities you have to get it approved through ARB. Once site approval and plan approval is done, then you can start your own function with the radiation safety officers and other things. You should have it, and you can start it. There is no problem. Now the things are open.
So that means it's this pharmaceutical company should be near to where the user end, that is near
No, not the generator we are sending across the country. We are production is done in Vashi, but we are Delhi, Kashmir, Kolkata, many places we are supplying the generators. Okay, production is because of the generator usage life is 10 days. So once you supply within 2 days, the third day onward, the doctor can use that the eluents and they can inject to the patient. Okay, formulate it and use it. So
that it is prepared at the time of administration.
Administration. It is that. That's what the advantage of generator. Otherwise, 6 hours we cannot use it.
Okay. Sir, we have another question from Shah. She is asking, what are the acceptable RCP limits for radiopharmaceuticals?
This actually I wanted to put the specification of because most of the radiochemical purity is more than 90%. 90 to 95% is sufficient. 92, some cases 90, some cases 95. It varies from the, that's why I have not put. Only typical thing I thought of putting it, maximum 95%. You put it, the 95% will be ideal. Okay. Radiochemical purity.
She is further asking that do all the batches qualify QC test? If not, what is done with rejected batches?
Well, see, our experience with the thing is most of the cases, 1 or 2 failure happens. What she was telling is absolutely very good question, because whenever some batches, when compared with the pharmaceuticals, here the difference in batch size is very small. Suppose the weekly production is there, daily production is there. Weekly production means suppose 100 consignments will go. Out of 100 consignments, what is the thing is 100 consignments will be wasted. Whereas in the case of pharmaceutical, if you prepare the manufacturing of tablets, it will be lakhs of tablets they will be manufacturing. Lakhs of injectables they will be manufacturing. Example, now you see the Covishield, this one, how much crores and the doses they are manufacturing. Not like that. In the case of radiopharmaceutical, the production is faster. That is short-lived. Weekly production is done, and the wastage will be 100. Suppose 100 vials will be wasted, then 100 vials we will allow it to decay. In the case of 19, according to our experience, that batch will in a year 1 or 2 batches fails. Where it fails, I will tell you. Some cases pH is failing. Any case it fails, pH is failing, we reject it. And the case of some of the coloration is found. The colorless compound, it should be there. Coloration, even coloration is there, we will reject it. So that case, then we will allow it to decay and we will dispose it. It is not much waste. Since the weekly production is there, it will take care. Otherwise, what happens? Just like pharmaceutical, once in a while you are producing hundreds of thousands, lakhs of tablets or lakhs of injectables. You produce it. Once it is failure, 1 lakh injectable will be thrown. No, it is not like that in this case. The waste is minimal. But only thing is radioactivity part we have to take care and properly we have to dispose. That is the only
Then sir, about the package, means once you withdrew your whatever the dose is there, and now the empty vials or whatever is there, how do they
empty vials? Empty vials only we will keep it aside and we will allow it to decay and we will dispose.
So that is also done. Means you have to decay those packaging also.
Oh no, not packaging. Packaging will not pack. First, if it is failed, we will not pack only. No. Okay. Once the QC reports is out, then only we will pack it, except a sterility test.
Okay. Sir, we have a question. Yeah, sorry. Yeah, continue.
Yeah, ask me.
Okay.
Hello.
Yeah. Dr. Sunil Reddy is asking how to do molecular docking of organometallic and radiopharmaceutical substances.
Organometallic substance and
radiopharmaceutical substances. Molecular docking.
Molecular docking. This particular thing I don't have much idea because nobody has come forward with this idea for molecular docking with radiopharmaceuticals. So this I will have to inquire and I will let you know the thing. Okay.
Asking about stability of astatine is very less. So how we can increase the stability?
No, stability astatine, that half-life is there. You cannot increase the half-life or decrease the half-life. That is the constant. That we cannot do it. That we have to formulate certain formulation only we can help to. It will not be half-life cannot be or shelf-life cannot be. Cannot increase. Okay, of the radioactive materials. Okay. That is the inherent property. No, that thing we have to. That's why the different isotopes have got. See, so many isotopes, medical isotopes are there, radioisotopes. So we are. That's why see the iodine. Why we are not doing only with iodine? Why we are going for technetium? Why we are going for the gallium? Why we are going for F18? Why we are going for phosphorus 13? Because each radioisotope got the advantage, disadvantages. One is having the long half-life, one is having the low activity, one is having the low radiation, one is having the detection limit is there. Like that, so many are there. Special activity, some compounds will get radiopharmacy will get more, some of the radiation radioisotope will get less. So each everything, what type of studies you are going to carry out, first you should be concept should be clear. Then selection of isotope will come in the picture. Then you have to go about your research. Like that, just like that, you cannot take any isotope. Okay, I like this acetine, so I will take it. So I cannot do like that. That day, see the what is your what is the you want to study. Concept should be clear. Then select the isotope accordingly.
Okay, so
Okay.
now Dr. Vaishali Shirsat, she has raised her hand. I think she
Okay.
would.
Okay.
Yes, sir. So yeah, actually my question to you was I wanted to understand more in terms of that what are the different isotopes which can be used for studying, with respect to the bone density or something like that.
Bone
Are there-
density.
Yeah.
Yeah, bone density. I think that phosphorus, that orthophosphate, one can think about it, orthophosphate, because it's a bone seeker. Okay. And
Okay.
now another one is there, MDP. One MDP labeled with technetium, that is methylene diphosphate. That is also goes to bone. Because for the bone imaging, we are using methylene diphosphate labeled with technetium 99. Probably that density thing one can study the bone density. Very good thing that I will also discuss with my person who was producing the thing. Bone density, which one is better? I will email to you. Definitely, I will mail to you. As far my knowledge is concerned, MDP, that methylene diphosphate, which is used for the bone imaging, one can use it. Even for the treatment of some of the bone palliation, that the orthophosphate we are giving injection, that also we can think about it. Some samarium based radiopharmacy, some EDTP is there. That is also one can think about it. 3 pharmaceuticals are there. Out of 3 radiopharmaceuticals, which is better, I will discuss with my friends and let you know. Okay.
They are available commercially?
Yeah, it is available with Brit. It is available with Brit. You can take it up. Yeah, it is available. But you cannot study at the Bombay College of Pharmacy because the radioactive handling, you require permission, and the waste degeneration, all the things are very difficult to handle. The radiation, what is the safety precaution may not be possible to take in your laboratory. You will have to go to Brit and study. Collaborate with the people. You have the idea, you concept, you develop it, and you take for the funding. You will get around you can get around 20-25 lakhs easily. You can get funding from BRS. That you can study. It's good study. Good thinking.
Okay, sir. Yeah. Thank you, sir.
Yeah, thank you, sir. Such a very informative session, and I think our participants they must be satisfied with answers which they have for the
Hello.
questions they have raised. Thank you very much, sir. Now
Okay.
over to Sagar for further.
Yes.
So,
Thank you, Dr. Ujjwala Shinde, ma'am, for chairing this session and also accepting our invitation on such a short notice to chair the session. Again, I would like to thank on behalf of the organizing committee. Now I request Dr. Vaishali Shirsat to present vote of thanks to the speaker. Over to you, ma'am.
Thank you, Dr. Sagar and Dr. Ujjwala. Thank you so much. Yes, indeed, it was a pleasure listening to Dr. Padmanabhan sir, who has highlighted about the radiopharmaceuticals quality control, which in fact is a very, very important factor, as you can see with the questions also which were raised by the participants. So, as you know that during drug metabolism studies, these radiopharmaceuticals actually play a very important role in order to understand the biodistribution of your molecule. So, which can easily be studied by the radio imaging techniques, and that's where the radiopharmaceuticals play an important role. So, thank you very much, sir, for enlightening about these parameters to us. And as a token of appreciation and in acknowledgement for this, your contribution for this faculty development program, our institute students and the housekeeping staff have actually planted a sapling on your behalf. So I
okay, thank
request.
you.
So
Yes, thank you.
I request the team to please play the video.
Okay.
Hello. Yes, sir.
Thank you very much for this nice, wonderful gestures by you, Bombay College of Pharmacy, that organizing committee. Thank you very much. And here one suggestion that whatever your plantation scenic, and my suggestion, humble suggestion is when you plant it, you can make a herbal garden. Any speaker is coming, one herbal plant should be planted. That will be a very good for your pharmacy college actually.
The tree that we have planted for you is the fig tree, and we do have medicinal plant garden. In fact, for the pharmacy institutes, it is mandatory to have the medicinal plant garden. So we do have it in our college.
Okay, thank you very much, Dr.
Thank you, Dr.
Thank you. Okay, it's nice.
Thank you, sir.
Thank you, Vaishali ma'am. We all will break for lunch now, and after the lunch we have one more session, that is session 9 of day 3, on day 3. So we will join back again using the same link sharp at 2:45. Thank you.